Hello students, welcome back to my YouTube channel. This is Dr. Sumedh. Today tutorial I will discuss about meiosis. This is very important lesson in cellular basis of life. That is second lesson of the A-level syllabus. We have discussed about cell division and mitosis. In this tutorial I will discuss about meiosis. Meiosis is important in sexual reproduction. It is happening in sexual reproduction and it's a different type of cell division, and there's a particular type of nuclear division which is happening in specifically in sexual reproduction. The results of the meiosis is four haploid and genetically non-identical daughter nuclei which is coming from diploid mother nuclei. Yeah, daughter and sons, this is the thing. We have four haploid genetically non-identical daughter nuclei, and in the mitosis it is identical to diploid cells which we have received in the mitosis. And in the meiosis there's two consecutive nuclear divisions happen. First one is meiosis 1, and there's a reduction division. That means there's a reduction of nuclear material. And meiosis 2, it is similar to mitosis. If we take meiosis 1 and meiosis 2, each of consists of four subphases: prophase, metaphase, anaphase, and telophase. According to the, if it is meiosis 1, we call them prophase 1, metaphase 1, anaphase 1, telophase 1, and if it is in the meiosis 2, we name them as prophase 2, metaphase 2, anaphase 2, and telophase 2. So before the cell division happened, in the S phase, the part of the interphase, DNA replication happen, and we will have two N, that is diploid cells, diploid mother cells we have. And at the first part of the meiosis, that is meiosis 1, if we take like this in this figure, if we take two chromosomes, one chromosome will go into one cell and the other chromosome go into next cell. So there's a reduction of nuclear material in the meiosis 1. After the meiosis 2, we will have four haploid cells which are different to each other. These are, we call them non-identical. So let's discuss about this process. Meiosis 1. Before the meiosis 1, cell is in the interphase. So we know that in the S phase of the interphase, DNA replication happen, and this is, as we know, this is a reduction division. So first stage or the first phase of the meiosis 1 is prophase 1. So there's a condense of chromosome, and nucleolus will be disappear, and nucleolus breaks, and centrosomes move to the opposite poles, and the cell is 2n. The homologous pair of chromosome will be paired in this situation. And there's a specific situation happening in prophase 1. This is very particular to meiosis, and it is happening in the prophase 1. This is called synaptonymial complex formation. What is it? Synaptonymial complex. There we have pairing of two homologous chromosomes. The two homologous chromosomes, the homologous chromosome pairing happen, and you name them as synapses. The initiation of chromosome pairing is there, so the two homologous chromosomes come together, and non-sister chromatids of the two chromosomes will be having crossing over. This is called crossing over. So each chromosome chromatids will be breaks and exchange and rejoin at the corresponding point of the other chromosome. And the crossing over places, the point visible where the crossing overs happen, you call this chiasmata. And when the kinetochore is formed, this, there's a separation of two chromosomes with the new chromosome formation. So you call it a recombination. So the genetic material will be changed. As this figure shown, as shown in this figure, you have blue chromosome in one side and the other one is the purple one, and ultimately we will have one part from the blue chromosome, that is chromatid part, and in the purple one there's a one small part of the blue one, and blue one there's a small part of purple one. So there's a exchange of chromosomal nuclear material. This is very important in variations. So because of there's a recombination of chromosomes, there's different characters formed. This is very important in meiosis. This is you call synaptomimeal complex formation. In the metaphase 1, the homologous chromosome arranged at the metaphase plate. These are arranged and arranged randomly. So whether it is coming from mother side or father side, it is immaterial. So the two chromosome pairs will arrange in the metaphase plate or in a random way. So in this phase, chromosomes each attached to the microtubules with the kinetochore microtubules to the opposite poles. Right. At the anaphase 1, the homologous pairs will be separate. Homologous pairs will be separate to the both sides. And one from each pairs move towards the opposite pole. But this is very important. Sister chromatid remains attached to each other. That means there's no separation of chromatids, which are genetically different. That is very particular in meiosis. This is representation of anaphase of mitosis and anaphase 1 of meiosis 1. In the anaphase, we know that there's a separation of chromatids. But in the anaphase 1 of the meiosis 1, we have separation of chromosome, which is from homologous chromosome pairs, which have been coming from one from father and the other one from mother. As a result, we have N cells. Previously, we have two N cells at the start, and we will have N cells. Two N cells will become N cells. So you call it nuclear matter reduction. Nuclear matter reduction. There's a reduction division. And telophase is the last stage of this nuclear division, and we will have haploid chromosome. A nuclear envelope reforms, and we will see the nucleoli reappears, and the spindle will be disintegrated. As a result of this nuclear division, now this in the telophase 1, we will have haploid 2 nuclei. Haploid 2 nuclei. In the cytokinesis, it is simultaneously happening with the telophase 1. So as we know, animal cells cleavage furrow is formed, and then the plant cells we will have cell ferrate. So as the results, two genetically non-identical, but it is haploid daughter cells will be formed. There's no DNA replication between meiosis 1 to meiosis 2. And after this stage, the cell will go into second consecutive division, that is meiosis 2. That is the second part of the meiosis. And the result is genetically different 4 haploid cells. So prophase 2, again chromatin will be condensed and chromosome with chromatids. So we can see in this figure, because of that crossing over and exchange of nuclear material, chromosome parts, we will have different chromosome in each cells. And spindle start forming 2 cells, nuclear annula breaks and nuclear disappears. Metaphase 2, and all chromosomes will be attached to microtubule. And at this point, one chromosome will attach to both sides microtubules. And at the centromere. So there's a as we discussed previously, there's a crossing over of 2 sister chromatids. So we have non-identical chromosomes. So chromosome will be at the metaphase plate, and it is very important. This metaphase plate is perpendicular to meiosis 1 metaphase plate. Previously we have metaphase 1 at the meiosis 1. That metaphase plate is perpendicular to new 1. And this point we will have 2 cells. And the other one is microtubule attached to kinetochore at the centromere, and it is attached to both sides, from the both sides to the one chromosome. Anaphase 2, and this stage sister chromatids will be separate, and these chromatids will move to opposite poles. And telophase, nuclear and nuclear reform, and there we saw the spindle is disassembled. At this stage, we will have 4 nuclei. These nuclei genetically non-identical. That means genetically different. We will have genetically different, and the specific thing is here you have haploid nucleus. Haploid nucleus. It is cytokinesis. It's also like mitosis again. So in animal cells, there's cleavage furrow is formed, and plant cells it is cell plate, and we will have 4 cells. Ultimately, we will have 4 genetically non-identical, and it is haploid cells. This is the representation of mitosis and meiosis. We can compare two in the meiosis as the figure A part. So uh, in the S phase, there's a replication of DNA, and meiosis part, yes, you have the 2 cells, cell divisions. Right, in the meiosis, synthesis of DNA replication, synthesis at the S phase, that is DNA replication, and with the prophase 1 especially, just recombination, and at the meiosis 1, chromosome separation, and you will have 2 cells with N, that is not 2N, that is N cells, and ultimately you will have, with the meiosis 2, you will have 4 N cells, 4 haploid cells. These are the comparison of mitosis and meiosis. The results of the meiosis is 2 identical cells. So mother cell is similar to daughter cells, and you will have 2 identical cells. In meiosis, it is non-identical, so that means genetically different, and haploid. These are haploid cells, that means 1N, not 2N. And this is important in sexual reproduction. Why? The other cell is coming from the opposite sex. So these are the significant of meiosis. This will maintain the constant number of chromosomes. So in the sexual reproduction, we will have two opposite sex cell coming from two opposite cell. So if the meiosis is not happen, what will happen? There's a doubling of chromosome number. But because of this is reduction of chromosome, that half it become half at the meiosis. So thus we can maintain the constant number, that number, rather 2n ultimately, because haploid cells, two haploid cells will come together and form the zygote, and this is 2n. So in meiosis there are different genetic variations. Why? Because of the crossing over happening and the separation, there are various genetic characteristics. These are going from one generation to next generation, and this will result in evolution. So these genetic variation is due to crossing over at the meiosis one, also with the recombination and the chromosome assortment. You call it independent assortment. That's mean the chromosome coming from mother side and father side will be assorted at the metaphase plate. So it is independent. So you will have different characteristics and results in genetic variation. This will leads to evolution of the species. So the end of the tutorial about meiosis. Thank you.